Awesome.
Hello, and
Should I
thank you
just
for joining us. I'm Kristen McPike, Program and Alliance Manager of the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Serge Biswas, will be discussing the Novo design of epitope-specific antibodies against soluble and multi-pass membrane proteins with high specificity, developability, and function today. Please note the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Without further ado, I'll turn the show over to our speaker.
Great. Yeah, thanks so much for having me. Really appreciate the opportunity to present to everybody. Yeah, really excited to talk to you all about some of the work we've been doing at Nabla on de novo antibody design, and sharing some results and the vision we have for how generative AI coupled with wet lab experimentation can make an impact on the design of antibody therapeutics. So I thought I would maybe just start with uh, a high level overview to the company and just talk a little bit about our mission at Nabla. And that's to expand the scope and efficiency of biologics engineering using integrated AI and experimental approaches. So when I talk about efficiency, I mean decreasing the time or cost it takes to run discovery and lead optimization. And in terms of scope, I mean increasing the set of interactions or pathways that we and pharma can feasibly drug. And sort of the concrete vision here is, uh, ultimately it'd be great if we can develop AI and experimental approaches such that we could, simply just generate a few dozen designs, and that most of them would actually be binding our target protein of interest and have excellent developability profiles, and not just bind the target of interest, do so with a high degree of atomic precision. If we can unlock that would allow us to directly be measuring performance of our designs in the most clinically relevant settings possible versus working them through earlier stage proxy assays. So one of the key bottlenecks we see in allowing us to achieve this mission is atomic precision. So we believe epitope-specific design is going to be a huge unlock in drugging difficult targets and pathways. So probably don't need to explain this too heavily to this audience, but some examples of problems that are hard or challenging to solve with traditional discovery are ones where we need just a high degree of atomic precision or epitope specificity to achieve the therapeutic effect we care about. So um, this would include, for example, blocking protein-protein interactions, or in some cases, inducing protein-protein interactions. In many cases, we're trying to bind a specific member of a target family, and we want to have highly selective binding. Or in some cases, we actually might want to be binding multiple members of the family, but not others. So being able to tune selectivity or engineer polypharmacology would be a very helpful skill. And then for a lot of the targets that we're especially interested in at Nabla is being able to engage very limited epitopes on the surface of targets. So for some of the most challenging targets like multipass membrane proteins, which include proteins like GPCRs, ion channels, transporters, there's typically very little extracellular real estate that's available for binding. And so to the extent we can develop AI approaches that can very precisely bind those limited and few epitopes, could be a major advance in allowing us to unlock those difficult target classes. And lastly, this is something we're still quite far from, but for many targets, proteins are not static things. you know, them adopting different shapes and conformations is often very important to their biology. And if we can have methods that allow us to be precise in terms of the conformations of different proteins we can drug, that would also be very enabling for a number of different therapeutic applications. So at Nabla, we've been developing a generative modeling pipeline, which we call JAM. And this is not something we have been discussing the methods behind, so I'll focus a little bit more on the input and output of this model. But before I sort of dive into the pipeline itself, just want to define what I mean by de novo design. And basically what I mean is, given just the target sequence and potentially the structure of the target, and without providing information of any known antibody binders, can we generate a new antibody structure and sequence completely from scratch to bind that target protein, ideally at a specified location on that target. So JAM is a generative modeling system that we've developed that allows us to achieve this goal. And in terms of input, what JAM will take in is a target sequence and potentially the structure of that target, as well as user-specified epitope residues on that target shown in dark green. And then what JAM is trained to do is look at this as an unfinished protein complex. And given this unfinished protein complex, JAM will essentially fill in the blank or finish the protein complex by generating an antibody structure and sequence it thinks it's going to engage that target at the specified epitope residues. So to date, we've worked on a variety of different targets, and broadly, I'm going to categorize these into soluble and multipass membrane protein targets. Throughout the talk, when we talk about multipass membrane protein targets, I may abbreviate them as MPMP. And I'll also sort of organize the talk in terms of targets that are maybe easier, at least initially, and then sort of progress our way through increasingly difficult and more challenging targets and design challenges. Also try to focus on examples where epitope specificity has been key for not just binding the target in a therapeutically relevant way, but also achieving certain functional outcomes. And then also just want to refer folks to a paper we recently published on the results that JAM has been able to achieve on a variety of different soluble and multipass membrane protein targets. A lot of the slides are talking through figures that we have in the paper, and this has been covered in the press as well. All right, great. So I'll get started talking about some of our work on soluble targets and the design of VHH and paired antibody binders to these targets. So first target I'll talk about is the receptor binding domain of SARS-CoV-2. Just wanted to throw up an initial visual to illustrate sort of the diversity of de novo VHH designs that JAM makes to this particular target. So to orient you here, the target, which is RBD, is in light green. The epitope we're trying to target is in dark green, and the orange ribbon structures are example generated VHHs made by JAM, and just wanted to draw your attention to just sort of the diversity of structures that we can get from JAM, but all broadly targeting the dark green epitope of interest. So throughout the talk, I just kind of want to lay some groundwork in terms of the experimental workflows we'll be using. The first workflow I'll talk about is just our binder identification. and characterization workflow. So typically throughout the talk, the way we're setting things up is we are generating somewhere between thousands to up to 1 million designs, VHH or SCFE designs with JAM. Given those designs, we will clone them into a yeast display library, and then using yeast display with FACS, sort the population or the subpopulation of binders to our target of interest. We can then, once we have that sorted population, we can isolate the DNA, sequence it with next generation sequencing. And based on the sequencing reads, we can identify which of our original designs are actually showing up as binders, and we call those identified binders. So that's the binder identification workflow. And then once we have a short list of identified binders, again, really using yeast display here, we can take these designs and reproduce them in their therapeutically relevant format. So for VHHs, these will be as VHH-FCs. For SCFE designs, we produce them as full-length IgGs. We express them in an ex vivo-based system, purify the protein, and then characterize their affinity, a few different measures of early stage developability, including their production yield, polyspecificity, and monomericity. And then depending on the target and application, we'll characterize functional properties of that protein on cells. So starting with RBD here, we started with an initial set of 11,000 designs, went through our yeast display and FACS workflow, sequenced binders with next generation sequencing, and in this first round of design, we're able to get 65 binders that are binding at a 1 micromolar concentration on the surface of yeast. And of those 65, 23 of them are binding at 100 nanomolar antigen concentration on yeast. So we produced a subset of these as VHH-FCs and characterized their KD to RBD by BLI. And we saw that 3 of the 20 actually had double-digit nanomolar affinities. Most of them had triple-digit nanomolar affinities, and a minority of them were micromolar binders or worse. Just to orient you on the plot here, the various bars in this plot are each a different design that we made, and the y-axis is the KD or binding affinity, lower being better. So we took those top VHH designs, and again, given we were targeting the ACE2 binding site on RBD, wondered if these were functional neutralizers of pseudovirus, and indeed they were. So if we look at our top VHH designs, these de novo designs showed, the best one showed picomolar, single-digit nanomolar neutralization efficiency to SARS-CoV-2 pseudovirus, which is pretty exciting to see, again, for a purely de novo designed VHH. And we next wondered whether we were actually binding the intended epitope. So short of solving the structure, another way we can assess this is with epitope mapping. So what we did was we looked at all surface exposed residues on the surface of RBD and systematically mutated each one to an alanine. And if we only focused on the mutations that didn't appear to disrupt protein stability or the stability of the target, and among those, look at which alanine mutants confer a loss of affinity to our de novo design, we found that there was 3 different alanine mutants that showed a greater than 5x increase in KD, meaning the binding affinity got worse. And all 3 of those alanine mutants co-located to where our de novo VHH design was binding. We also characterized all of the binding VHHs for early stage developability properties, looking at properties like production yield, percent monomericity, as well as polyspecificity as measured by BVP ELISA. And generally, these de novo VHHs looked pretty good. So in terms of production yield, over half of them are expressing at greater than 100 mg per liter purification yield. They're all, or nearly all, greater than 95% monomeric by SEC. And in terms of BVP ELISA, where lower is better on this third plot, the majority of these designs actually looked less polyreactive than trastuzumab, which is a well-established non-polyreactive clinical benchmark. Another reasonable question that folks have when looking at this data is, what does the sequence composition of these de novo VHH designs look like? And in particular, how human are they? So we looked at the humanness of these designs and saw that, so our metric for humanness here is what is the percent identity to the closest human germline. And the distributions you see on the left-hand side of the plot there just indicate the distribution of humanness values you see among clinical stage antibodies. So if you look at, for example, chimeric antibodies that, the V domain, it comes from another non-human species, that's the orange distribution. The light green distribution is for humanized antibodies, and then the dark green distribution is for fully human antibodies. So you can see our designs, which are the black dots, sort of overlap with chimeric or humanized antibodies, which is lower than we would like. And so this is, at least at the time of these results, a limitation of the designs that JAMs are making. So showed some evidence that this works for one target, RBD. Question is, how does this generalize to other soluble targets? And what does the specificity of these designs look like? So we set up an experiment where we made 10,000 designs to 5 very different soluble targets, and pooled all 10,000 of those designs into a single 50,000 member library. We then took this library and screened it for binding against all 5 targets, as well as a off-target protein, ovalbumin, which has been shown to serve as a reliable polyspecificity reagent. So this pooled experiment allowed us to basically assess the specificity of our designs all in sort of one shot. And generating and screening designs to these 6 targets in parallel, it's worth noting that this was actually just marginally more work than doing one. So we think this is also another exciting direction for de novo design, is in the ability to parallel process multiple targets in one go. So these are the results, and what I'm showing you here is rows refer to the library from which the designs come, and columns refer to the target that we measured binding against. So what's shown in this heat map are all of the binding designs that we saw to any target among those 50,000 designs. So to walk you through this, if we look at designs targeting the extracellular domains of HER2, we had in total 21 designs that bound HER2, and 19 of those designs came from the actual HER2 set of designs. 2 of those designs came from designs we had actually made against PD-1. Similarly, for IL-13, we had 14 binders to that target overall. 13 of those came from the library that we made that were actually targeting IL-13. So generally what we want to see is a nice diagonal on this plot, and that's more or less what we see. Interestingly, for the designs we made against PD-1, we did observe some off-target binding. So most of the PD-1 binders are actually binding PD-1, but we do have a couple that are binding off targets, indicating some low level of lack of specificity. But another encouraging data point here to point out is if we look at binding of this whole library to ovalbumin basically saw no binders to this polyspecificity reagent. So this was encouraging evidence that the designs that JAM are making are reasonably specific, though there are some exceptions. So one of the interesting recent trends in the world of machine learning is this idea of applying more test time compute. So using more compute when you're applying the method to try to get better results. So in the world of large language models, this is corresponded to allowing these LLMs to essentially think and introspect on their outputs before sort of returning an answer to you. And if you're a ChatGPT user, this might be something you've seen and how you've interacted with the models. So it's a similar idea here that we can apply, where initially we start with just the target information. So again, just the target sequence or potentially the target structure as well, if that's known. And we use JAM to generate an initial antibody target complex. And before sort of going into the wet lab and testing those designs, in a fully computational loop, we can take those generated antibody target complexes, which we'll call the round one complexes, and feed them back into JAM. And essentially what JAM will do is take those as input and elaborate upon them to generate a new set of round two antibody target complexes. And then we can feed that back into the model again and generate a set of round three complexes. And the idea here was that, well, if we let JAM sort of continuously refine its outputs, maybe we'll get better de novo bind rates and maybe affinities as well. So to test this from each round of, again, fully computational introspection, we siphoned off 10,000 designs from round one, 20,000 from round two, 20,000 from round three, and actually measured binding of these designs to, in this case, the target was RBD, and asked these questions: what is the bind rate as a function of introspection round, and also what are the resulting affinities? So excitingly, what we saw was that the more we let JAM introspect on its outputs before sort of recommending designs to test in the wet lab, the better the hit rates become, and also the better the affinities we see. So in this plot, what I'm showing on the x-axis are the different rounds of introspection, so R1, R2, R3. And the y-axis is showing the binders we saw per 10,000 designs we tested. And then the different shades of the bars are the different target concentrations. And this is all binding data generated by yeast display. So you see in round one, for example, we see about 10 binders out of 10,000 designs binding at a 1 micromolar concentration on yeast. We compare that to round three, we see that nearly doubles, or more than doubles, to about 21 designs per 10,000 designs. But also very excitingly, is you can see we're starting to get binders at those lower antigen concentrations as well. So if we look at 10 nanomolar binders or 1 nanomolar binders, after 3 rounds of introspection, we're actually able to get more and tighter binders than we could without doing this introspection. Okay, cool. So we talked a lot about VHHs so far. The next obvious question is this method only specific to VHHs, or can it design paired antibody formats, for example, SCFVs and their sort of full-length monoclonal form as well? So in this case, had a similar binder identification and characterization workflow, but to walk you through how we do the designs for SCFVs, or at least how we used to do the designs for the SCFVs, and for all the results in this talk, we're actually designing 3,000 VHs and 3,000 VLs, and then we clone them combinatorially to get a much larger library. So 3,000 times 3,000 is 9 million total scFv designs. Again, we put that through use display and fax, sequence out the binders with NGS, and then given those identified binders, we'll do follow-up characterization on those. We attempted to design scFvs to RBD, and what we saw after going through this workflow is 155 binders that bound RBD at a 1 micromolar concentration on the surface of yeast. And we took 19 of those, a random 19 of those, and 10 of them showed double-digit nanomolar affinity for RBD or better. And so that's on the left plot here. Again, y-axis is KD. The x-axis is just the ordered design. You can see, about half of those tested were double-digit nanomolar or better. And then we took all of those and characterized them for pseudovirus neutralization. And excitingly, we saw about 6 of them look like they were neutralizing pseudovirus with picomolar IC50s. And unlike the VHHs, the humanness of these design maps were substantially better. Essentially, if we look at the VHs, these tended to have humanness scores consistent with that of humanized antibodies. So not fully human, but better than chimeric clinical stage antibodies. And if we look at the VLs, these had percent identities to closest human germline exceeding 80%. And again, were consistent with that of humanized antibodies. So some initial takeaways here. Epitope-specific design allowed us to act on a strong structural hypothesis for how to achieve neutralization for, in this case, a well-understood target. So in this case, for RBD, we were targeting the ACE2 binding site, which is important for coronavirus's infection process, and disrupting that interaction with the human ACE2 receptor. It seems like a good way of disrupting that. That's been shown many times. So we could really act on that with de novo design. And from relatively small libraries, so in the VHH cases, looking at roughly 10,000 member libraries, and in the scFv case, looking at million member libraries, still relatively small compared to larger naive libraries, which might be 10 to the 11th in size. We were able to obtain epitope-specific VHHs and NAbs that had double-digit nanomolar affinity, and several of these were picomolar neutralizers of pseudovirus. Generally speaking, design showed good early-stage developability properties. Developability is a large and complex picture. So of course, there would be a lot more that would need to be done to show these are actually developable. But according to these early stage metrics, these designs look pretty encouraging. And one current limitation is that the humanness of these designs need to be improved. And that's something we've been working on. Lastly, two more points to make here. Most of what I showed you was targeting RBD, but we did show that jam can generalize to other soluble targets and can design to other targets in a relatively specific way as well. And then one of the most exciting results we had at the time was showing that by allowing jam to reflect or introspect on its outputs before recommending designs for testing in the wet lab, we could significantly improve bind rate and affinities. And slightly academic point, but it's actually the first demonstration of inference time scaling is applicable to design of like an actual molecular system. So we're very excited about that and are continuing to explore this. Okay, so those were soluble targets, and another exciting question is whether de novo design can be used to generate binders to more challenging targets, particularly multi-pass membrane protein targets. So again, these include targets like GPCRs, ion channels, tetraspanners, transporters, so on. And what's been limiting for these targets is that they have very little of their total protein surface area available for binding. So these are targets that are typically heavily embedded in the cell membrane, with most of their protein surface area either making contacts with cell membrane lipids or actually below the cell membrane, and a small minority of the protein being available for binding. Another challenge here is with many of these targets, they have very closely related family members. So often we want to bind one member of the family, but not have binding to other members. So target selectivity is really important. And then a lot of these targets are conformationally dynamic and exist in active and inactive states. And it's not something we are going to show in the deck today, but as an application of generative design, if we can target specific conformations of, say, a GPCR, maybe the active state or inactive state, that could be very enabling for making therapeutics. The last thing I'll mention here is these are sort of not a niche set of proteins. Multipass membrane proteins actually comprise 2/3 of all cell surface receptors, but are actually targeted by less than 10% of approved biologics today. And that's really just a reflection of how challenging they are to generate binders for. Okay, so first target I'll talk about is Claudin-4. This is a tetraspanner, meaning it's got 4 multipass alpha helices that are embedded in the cell membrane, and sort of the dark green residues you see in this graphic here are the extracellular loops. And this is just an example of a jam-generated VHH that is engaging these extracellular loops, or I'll just refer to these as ECLs throughout the rest of the talk. So for these targets, again, the fact that they are membrane bound makes it hard to screen for binders against. And essentially the challenge is, because these targets are membrane bound, you can't separate them from the context of cells. So when you're screening for binders, in many cases, you need to bring the whole cell along with you, or come up with a way of separating this target from that membrane context through some type of solubilization. And there have been many different approaches that have been explored for solubilizing these targets. But a new idea we explored here was, well, we know JAM can help us design antibodies, and we've actually trained JAM to be a general purpose generative model over proteins. Could we use JAM to generate solubilized versions of a difficult multipass or MPMP target? So what we did here was we took the native Claudin-4 sequence and structure. We gave that as input to JAM. JAM generated 10,000 de novo VHH designs targeting the ECLs of Claudin-4. But we also used JAM to generate a soluble proxy of Claudin-4. So in this case, the way this works is we take that native multipass membrane protein target, again, Claudin-4 in this case, we generate this soluble proxy where what JAM is trying to do is, while preserving those key extracellular residues and structure, sample a new protein scaffold to essentially support and preserve the structure of the ECLs. And this scaffold is completely distinct in sequence and structure to the membrane spanning helices of Claudin-4. And that scaffold itself is designed to be a nice, stable, soluble scaffold, again, to support those key extracellular loop residues. So given the soluble proxy, if we can succeed in designing one that has high fidelity to the native target, we can use it as a screening reagent. So At this point, we can use our normal yeast display-based workflow to screen our de novo antibody designs against this Sol Claudin 4 proxy. And then once we have a short list of binders, go and measure which ones are actually binding Claudin 4 in its native form on the surface of cells. So these are just some FACS plots showing you that working with the Sol Claudin 4 proteins is actually just very similar to working with any other soluble recombinant antigen. So we made our de novo library in the first round of yeast-based screening. You can see sort of a distinct binding population here, which we can sort and enrich, and in the second round, get a more enriched population of binders to this Sol Claudin 4 antigen. We sequenced that with next generation sequencing and observed about 50 Sol Claudin 4 binding VHHs. We took all 50 of those and characterized which ones are actually binding native Claudin 4 on the surface of cells, and characterized binding to two different cell lines. And excitingly saw actually 3 of them showed binding to native Claudin 4. So in the first plot on the left, I'm showing you binding to an overexpression cell line, as well as binding to a target negative parental cell line. So you can see 3 of our binders here. Again, completely de novo VHHs are binding native Claudin 4 with double-digit nanomolar binding affinities on the surface of cells with no binding to the background cell line. And then we also verified for our top design that it is binding a disease-relevant cell line that's known to express Claudin 4 endogenously. So in these OVCAR3 cells, we also saw a similar binding profile and an isotype control here was negative, suggesting these designs are actually binding Claudin 4. Excitingly too, for the claudins in particular, selectivity is really important when it comes to making therapeutics against these targets. So we characterized the specificity of these de novo designs to closely related off-target claudins, so claudins 3, 6, and 9. And we saw that all 3 showed greater than 100x selectivity for Claudin 4, which was the intended claudin over these off-target claudins. So in the top left plot here, the binding profile for design 1 overlaps with that of a positive control antibody known to bind Claudin 4 specifically, and the light gray distribution here shows unstained cell control, basically indicating what non-binding would look like. And we can see that our design 1 is not binding Claudin 3, 6, or 9. And we repeated this characterization for those 3 Claudin 4 binding designs and saw a similar result across the board. Okay, wanted to share an example of another claudin. So this is a undisclosed claudin, but different from Claudin 4. And the goal here is not just to get binders to this target Claudin X, but we want to have no binding to a very, very closely related off-target claudin, which we'll call Claudin Y. And one thing I would just encourage you to think about as we're thinking about this is there's many such examples where this kind of selectivity is important. One example is for 2 closely related isoforms of a target, or between 2 highly homologous members of a particular target family. So yeah, just to sort of illustrate what I was saying in words, Claudin X and Claudin Y are 2 closely related claudins that historically have made it very challenging to obtain Claudin X selective binders. And that's really, again, because there's only a handful of amino acid substitutions that differentiate Claudin X and Claudin Y in their extracellular loops. So we took a similar approach to what we did with Claudin 4, but in this case, we have sort of 2 claudins very intimately in the picture. So we took native Claudin X and native Claudin Y. These went into JAM separately. We used that to design a de novo VHH library against Claudin X. And we also use JAM to generate sol-claudin proxies of claudin X and claudin Y. So for sol-claudin X, we actually generated two different soluble proxies. I'll get to why that's important in a bit. And then we also generated a sol-claudin Y proxy. So given those, we screened for sol-claudin X selective binders via yeast display using all three of these proxies. And then once we had a short list of binders, actually measured claudin X selectivity on the surface of cells. And then I'll show data for, we took that information, used JAM to essentially propose a follow-up round of designs to improve selectivity even further. So just to illustrate some data around what these sol-claudin proteins look like, basically when we're making these, we use JAM to propose a couple dozen different designs. So in this case, we did about 24 designs of sol-claudin X, and we characterized a couple different things. So again, the goal here is we want a soluble proxy where the extracellular residues of the native protein are preserved both in sequence and structure, and the model is able to successfully generate a supporting scaffold that allows us to stabilize and solubilize the protein, but again, otherwise preserve the key extracellular structures. So as evidence, we've succeeded in that goal. We characterized three different things. How well does this soluble proxy express in a mammalian production system? What does the monomericity of that look like? And if possible, does this soluble proxy bind known conformational binders of the native target protein? So if we look at all of those properties here, if you were to try to take just the wild type claudin X and express that in a CHO-based system, as expected, you basically see no expression. Whereas for our sol-claudin X designs, the majority of them express with greater than 50 mg per liter yield in ex vivo, with some of the top designs, getting above 150 mg per liter yield. So just to summarize that, of the 25 designs, 23 of them express greater than 25 mg per liter in ex vivo. We characterized all of these 23 expressors for binding to a known conformational claudin X binder. So in this case, this was a monoclonal that is known to bind a conformational epitope on claudin X. And 13 of those 23 sol-claudin X designs we made are binding with a KD of less than 5 nanomolar to this known conformational binder, which is good evidence to us that we've preserved those key extracellular residues. And if we took a look at this top expressing design that's also binding this conformational binder and just run it on SEC, we see it's about 87% monomeric. And so sort of our takeaway from all of this data is that this soluble proxy seems to be relatively well biophysically behaved, and we have evidence that the ECLs are reflective of the native ECLs. And just to give some intuition for how we were using JAM to generate the de novo VHHs here, essentially we were guiding JAM to target residues that differ between claudin X and claudin Y. This is an example of one of the generated VHHs and how it's engaging the claudin and some of the residues that differ between X and Y. So we took those 50K VHHs and through a series of MACs and FACs, isolated a population of binders that were binding sol-claudin X, either design A or sol-claudin X design B. So the difference between design A and design B is the scaffold regions of these two sol-claudin proteins are completely different. So if you have a VHH that is binding both of them, it's reasonable evidence that it's binding a shared structure on both of the cell claudin proxies, and the only shared structure on the cell claudin proxies are those preserved ECL residues. So we saw about 128 of our de novo VHH designs are binding both cell claudin proteins. These seemed like good candidates to follow up on and see if they were actually binding native claudin X on the surface of cells. And so to summarize this data, so far we had 50,000 designs that we made. About 128 of them were binding the cell claudin, both cell claudin proteins. We characterized 86 of those. And of those 86, in a single point MFI screen, we saw that about 17 of them were binding the native claudin X on cells at 100 nanomolar VHH Fc concentration. And the big question here again was, how many of these, if any, were selective? So of those 17, we only saw 2 were selective, but still, that was pretty exciting that, purely de novo, we were able to get any selective binders at all. And so here are what their binding curves look like. So the first plot is for the first selective design. X-axis is just the concentration of the antibody, and the y-axis is just the binding MFI. And we can see that there's pretty good separation between binding claudin X and binding claudin Y here. And then for the second design, looks even better, get a much better Emax for claudin X binding relative to claudin Y. So this was exciting to us, but for this particular project, we were hoping to get more than just 2 selective binders. So we thought, okay, how can we improve on this result? And if you recall the idea of introspection, what we were doing there were purely computational rounds of introspection before testing anything in the wet lab. But we don't necessarily need to do that only computationally. So in this case, we have some initial experimental data that some of the complexes that JAM generated actually yielded selective binders. So can we just take those 2 selective binders we got and use JAM to elaborate new sequences and structures that are like the 2 original ones, but hopefully increase the diversity of structures and the number of selective binders we get at the end of the day. So using this elaboration procedure, we generated 100,000 additional designs. And using a dual labeling strategy where we first enriched for binders to cell claudin X, and then in a subsequent round, instead of labeling with just one antigen, we labeled with both cell claudin X and cell claudin Y as a sort of dual antigen labeled yeast. We sorted out yeasts that were only binding cell claudin X, sequenced those with next generation sequencing, and characterize which ones of those were actually binding on cells. So in this case, we made 100,000 designs. If we actually sequence this population of cell claudin X binders, there's actually 21,000 designs there that were binding cell claudin X. Of those 21,000, we produced a subset of 376 of those. And based on the on-cell bind rate we saw there, we extrapolated that about 14,000 of those 14,000 of the 21,000 are actually binding claudin X, native claudin X on cells. And then again, from the subset of 376 we characterized, we saw, based on the percentage we saw that were greater than 10x selective for claudin X, we extrapolated that about 1,000 of these 14,000 show greater than 10x selectivity for claudin X. And characterize the selectivity for some of the most promising ones. And these are just 4 examples of the top 4 most selective designs we found in this elaboration round. And you can see, compared to the previous designs I showed, we're seeing binding EC50s of similar in range. So these are still double-digit nanomolar binders of claudin X, but have essentially no binding to claudin Y, even when we titrate the antibody up to about 3 micromolar, which is a lot of antibody. So, after this, it felt like, especially for the top designs, we were able to get much more selective designs and get a bunch of them. In terms of sequence composition, what we saw was uh, uh, three distinct CDR clusters, and within each cluster saw a decent amount of diversity among the sequences. And we also looked at the immunogenicity, or at least a sequence-based measure of immunogenicity for these designs. So if we look at net MHC pan predictions of weak peptide MHC interactions or strong peptide MHC interactions among clinical stage approved antibodies, we saw that our designs had slightly worse scores than that of approved antibodies. So if we look at strong predicted peptide MHC interactions, approved antibodies typically have an average of 0.94, whereas our designs had an average about 1.82. And for weak interactions, about an average of 4.34 for approved antibodies, and slightly higher for our designs, average of about 6. So, these are roughly in distribution, don't look grossly more immunogenic, which we were excited about. Okay, and then very quickly, I'm running out of time, but also wanted to share that a result that we reported in the paper for trying to target the GPCR CXCR7 essentially went through a very similar workflow as we did for Claudin-4. So we made a Sol CXCR7 design. Top design we found expresses at about 20 mg per liter yield, and ex vivo is about 85% monomeric, and excitingly binds the native chemokine ligand of CXCR7, which is SDF1. So pretty good evidence that this is a good soluble proxy. We used it to screen 20,000 de novo VHH designs against Sol CXCR7. And of these 20,000, we saw about 32 binding CXCR7, and then measured binding against the native CXCR7 to see if any of those 32 Sol CXCR7 designs were actually binding the native GPCR. And excitingly, one of them did. And so that is to say the other ones didn't, but one of them did, and we're excited about that. And not only did it bind CXCR7, it bound with pretty good affinity. So purely de novo, we got a double-digit nanomolar affinity binder that binds native CXCR7. So doing a lot more work on the GPCR side. Hopefully can talk more about that soon. But some takeaways here is we can use JAM to not only generate antibody designs, but also well-behaved solubilized versions of more challenging multipass membrane protein targets. So we call these Sol MPMPs, and we've shown that they can be useful as antibody screening reagents. So this is the first demonstration of using Sol MPMPs to isolate new binders to a native multipass membrane protein. So we're very excited about that. And we could use JAM to also target limited extracellular epitopes on two fairly different MPMP targets, a Claudin and a GPCR. And as far as we know, this is also the first demonstration of a fully computationally designed antibody against a multipass membrane protein. For Claudin-4 and for Claudin-X, we observed excellent selectivity. Again, really important property to try to achieve for many families of targets. And with that, maybe some summarizing thoughts from the whole talk. Being able to design against particular epitopes was a key enabler for achieving functional goals for soluble target, especially RBD, and also generating binders to difficult multipass membrane protein targets. Just some inside or sharing some knowledge from the inside here. Getting here actually required a lot of collaboration between our ML and wet lab team. And actually the state of JAM and where it is now required over 50 iterations back and forth between our dry and wet lab teams. And in total, this comprised about 50 design build test cycles, where each cycle is approximately testing 10,000 to 100,000 antibodies, and we're updating the model based on that data. So in total, about 50 million internally generated data points. We are very excited about integrating JAM and these wet lab screening technologies because they're opening up new opportunities to generate and optimize antibodies to tackle soluble targets more efficiently or hit epitopes very precisely, and also open up target space generally by allowing us to go after more difficult targets. Really want to thank the team. So we're a team of, I think, about 17 people now, based in Cambridge, Massachusetts. We've actually just had 2 Adams join recently, so don't have their photos yet, but yeah, thanks to like a really talented, technically motivated team. And we're also fortunate to be partnering with some really great partners. We've signed collaborations with Takeda, AstraZeneca, and Bristol Myers Squibb, and we are interested in establishing partnerships with more companies. So if you're interested in working together, please reach out, bd@nabla.bio, and I'll take questions if there's time.
Thank you so much, Serge, and thank you, audience, for your attendance. This has been fantastic, and the participation. So we have a lot of questions, Serge. I don't think we're going to get through all of them before noon. So if you want to look at the Q&A section, and just if you could put like flags next to the ones you'd really like to answer, then I can read those aloud to you. But in the meantime, while you look for ones that you're really excited about, I'll start from the top. So question number 1 is, was your solubilized GPCR also mostly monomeric in SEC and expressed as well? Also, can you say what a scaffold has nothing to do with normal GPCR structure, or that it does?
Yeah. So yeah, this is one of the interesting things we've been discovering about these soluble proxies. So for the GPCR case, I forget the exact monomericity, but I think it was in the 70 to 80% range. So not super monomeric, and definitely for by soluble antigen standards, not particularly well behaved. But despite that, we've been able to use it to get actual native CXCR7 binders. This is not data I showed in the deck, but we'll have a lot more to say about this soon. We basically used that soluble proxy to get many more CXCR7 binders. So despite it not being super well behaved, still seems to be useful for screening. Yeah, I guess I'll take the next question.
Okay, perfect. Does your CXCR7 binder block ligand binding?
Yeah, again, this will be something we share more soon, but these do look like they compete for SDF1 binding, and are most of these are functionally antagonistic.
Perfect. Did you try doing pull-down experiments with lysed OVCAR3 cells to test the specificity further, or any other complementary experiments to confirm specificity?
We did not do that. The main thing that convinced us we were binding the target was just the overexpression cell line experiment. So we had an overexpression cell line expressing native Claudin-4, and then a clean KO, and it wasn't binding that KO cell line. So we felt pretty good based on that result. And then the OVCAR was just to show that it would generalize to another cell line.
Great. Okay. How are epitopes defined for soluble targets with significant surface area? And follow-up, what's the efficiency of identified clones that bind the epitope target?
Yeah, so this is not something we have tons of data around yet. Anecdotally, what we've seen internally, I think, reflects what others have reported in papers, in that generally we're trying to pick epitopes that have at least some degree of hydrophobicity in them. Again, this is somewhat more anecdotal, but polar, heavily charged epitopes tend to be harder for us to bind. But we haven't done a thorough systematic investigation yet. And there's also been a number of improvements we've made to the method, and we're not entirely sure if those improvements are allowing us to access more epitopes yet.
Thank you so much. So I'm going to ask a few more questions. I think we could probably extend to 12:05. With the continuous feeding strategy or iteration of antibody/AG structure complexes through JAM, notice anti-idiotype antibody outputs. Does that question make sense to you?
Just trying to read it. I'm not entirely sure what the question's asking.
Okay. We'll circle back to that one. Just trying to make sure I get one from each person who asked a question. So how about JAM creates the CDR loop sequences based on ML training sets of protein-protein interactions and then fills the rest with the framework structure of an antibody variable domain. This person just wants to make sure they understand correctly.
Yeah, so not quite. JAM is actually generating the entire VHH or scFv structure. So it's not sort of breaking it down in terms of CDRs and frameworks separately. That said, we have ways of guiding the model, especially when it's sort of doing the sequence generation, portion of the design, to adhere to a specific framework sequence. But I also do want to mention that it does seem pretty important for us. So for the results I showed you, it does seem pretty important that we are designing at least some of the framework to get binding. So if we design only the CDRs, the results are quite a bit worse. But if we design the CDRs plus some of the framework, we get results similar to what I showed you there. We've also made some improvements to the method where we're seeing much better success rates and affinities, and hopefully we can talk about that soon.
Thank you so much for answering all these questions. So I'm going to combine a couple of them because I think they're related. How big is the designed VHH library size? And what is the framework used to design the library? And how much of this work is published?
So the library sizes for VHHs, typically we're doing something between 10,000 and 100,000 VHH designs. There's no real limit on that. At some point, you're just paying for a lot of DNA synthesis. And the throughput of use display is so high, we would never sort of exhaust that throughput. But yeah, 10,000 to 100,000 designs is typically what we're doing. Increasingly as the method improves, the goal is very much to make that smaller and smaller. So we want to get to a place where we can make, ideally just 100 designs, and some tens of % of those are, just binding the target, and maybe we don't even have to use display. We can just directly make the proteins and measure whether they're binding the native target in its most relevant form. And sorry, could you just repeat the second part of the question?
How much of the work is published?
Yeah, so I think I shared the paper at the very beginning of my talk, but for most of the results I presented, we published a technical report, which I would definitely refer you to here. It's on bioRxiv and our website.
Thank you so much. Okay, I'm going to ask two more questions. The VHH binding to the undisclosed CLDN seems to bind through many non-canonical residues, i.e., it is on its side. Did JAM come up with designs outside of the CDRs and utilize framework sequences to come up with the solution? Yeah,
so the short answer is yes. For the designs that were successful there and selective, it was a combination of designing the CDR3, but there are several residues in the framework that are important for driving that interaction.
Perfect. Thank you so much. And then my final question will be, did you screen for agonists and antagonists? And also while screening, do you check functionality along with binding capacity?
Can you say that just that last portion again? You dropped out for a second.
Sure, no problem. Sorry about that. I said, do you screen for agonists and antagonists? And also while screening, do you check functionality along with binding capacity?
My answer to that question is stay tuned. So yeah, we'll have a lot more to say on that hopefully soon. And yeah, don't want to jump the gun on that yet.
Perfect. Okay. And I think that that's a perfect place to wrap up. So, Serge, I'll send you the rest of these questions because I still did not get through a good 6 or 7 of them. If you want to answer them, then alongside the webinar recording, we can share that information as well. So,
Okay, yeah,
I
sounds good.
want to thank you again for this wonderful presentation, and thank you to our audience for your great participation. This webinar will be available both on the Antibody Society website, on this Freestone platform, and on our YouTube page shortly. And if you have any questions, feel free to follow up with me at kristen.mcpike@antibodysociety.org. Thank you all so much, and thank you so much again, Serge, for this wonderful presentation.
Thanks, everyone.